the session further, and she's going to join tomorrow. Okay. Any doubt in the couplings?
No, ma'am.
Uh, can you tell me about the FPKM? Is it clear? Because that's the most important concept you need to know. So tell me if it's clear. Okay, fine. Let's go with our next step, that is cuff merge. So I will show you how to prepare the command. Is my screen is visible?
Yes, ma'am, it is.
Okay, so let's go with our next step, that is like once what is the cufflinks? Like what cufflinks is giving us the like what kind of output it is giving us. Can you tell me?
BAM files, ma'am.
Cufflinks. Uh can you tell me like up till where it's done like uh wait, uh sorry. Till here it's done, I guess so, right?
Yes, ma'am.
Hmm. So what is the output of cufflinks? Okay. Do you know the cufflinks is giving us these four files, right? Like out of that, this transcript.gtf file is one of the most important file, or this is the main important file of cufflinks output, which contains a count of gene expression, like how much amount of genes are expressed in a control one sample, then same in a control two, treated one and treated two. Means here we are getting the count of expression in separate samples, individual samples of genes, right? And what is a FPKM? Can you tell me? You can tell me, otherwise I will repeat it once for you.
It's fragment per kilo base per million.
Yes, it's a fragment per base kilomillion. And what it is giving?
So it's one of the it's a unit for normalization. Like there are different units for no after normalization. So like RPKM, FPKM.
Hmm. And do you know how it is calculated?
Uh, yeah, last week uh, she told us, Tanya ma'am told us.
Yes, yes. Okay, so it's done. Like, I just want to confirm that one. Okay, all right. So once this is done, like once we are having the normalized count of gene expression, but what is our main purpose in the RNA-seq? Is that too, we need to quantify the differential expression, right? We need to calculate the differential gene expression. And if you will see, like, as we want to calculate the differential expression means what we need to compare the samples, like how much the gene is expressed in a control one with respect to control two, or a so with respect to treated one, or how much the gene is expressed in a treated with respect to control. So to compare that, basically we need all the expression count together in one file, right? Are you getting my point?
Yes, ma'am.
So That's what we are going to be use the next step after the cufflinks, that is a cuff merge. Cuff merge means what? Like where we are merging all the samples together, all the gene expression count together. Because what the cufflinks is doing, so basically the cufflinks is removing the biasness which is caused by the factors like depth and the gene length, and then it is giving you the normalized count. So that normalized count we are merging together into a one file so that we can compare how the genes are expressed in one sample with respect to another. And that's what we are using the cuff merge tool. So if I will just open a manual of cuff merge, you can see that you have to just call the tool cuff merge. Then in -o, you need to put your output directory where your input file will be saved, like output file will be saved. Then in -small g, you need to provide your GTF file. Then in -s, you need to provide your reference sequence, and -p, that is for the threads. So let's prepare the command. But one more thing, what about the input files? Because how many input files we are having here now? As we have a four samples, we run the cufflinks four times, and we are having the four transcript.gtf file in the all cufflinks folder, correct? If you will see in the cufflinks control one here, this is what one transcript.gtf file we are having. Same file you will have in a control two, treated one, and treated two, which means like we are having a four input files, but it's not possible to give a four input file in a single command likewise. So that's what you can see here as per the manual. It is giving the assembly GTF list text file. Means what you have to do is you need to create one text file, and inside the text file you need to give the list of your GTF file, which means the transcript.gtf file. So what we have to do is, first of all, using the touch command, you need to create one file by the name of assembly, or you can give whatever the name you want, text. So if you will see this assembly.txt file is created, you can like normally manually also create it. You can create it. Then open this file, and inside that file, you need to put the path of the transcript.gtf, which means like what you have to do is you have to open the terminal from the cufflinks control one, then type the pwd. Then what you can do, you can just copy the path and then paste it over here. So now give the slash, and here if you will just do the ls, you have to give this transcript.gtf. This is a one path, which is a control one. Now what you can do, you can copy this path as it is, put it here, the next line, and we are having the cufflinks control too. So just it's a shortcut way to prepare the path. You can make it two. Then cufflinks treated one. Just make sure what is the name of the treated one. So it's a cufflink treated one. So in my case, treated T1 is capital. Maybe if you have some another name, you can give that as per your system, whatever you have given. Treated one, and then I'm just going to copy this as it is, paste here in the next line, and make it treated two. So this is how you need to prepare your input file for the cuff merge. Can you try?
And ma'am, what about the sorted control one? Like,
That's a BAM file. So that BAM file you used in a cufflinks, right? Here, if I will show you, that BAM files you used in a cufflinks already. That is a alignment file. So from that alignment file, cufflinks counted like how many reads are aligned to the reference genome at a specific position of gene, and that's what it will give you the count of expression. But that count of expression which is given by the cufflinks is a normalized count by removing the biasness caused by the depth and the gene length. Okay. Is it clear, sir? Or
Yeah, ma'am, got it.
Hmm.
Can you again show the text file which you have
Yes,
created?
yes. Yes. In your system, these names can be different. Okay, so just give according to your system.
Done, ma'am.
Hmm. What about others?
Just one.
Yeah, take it.
We need to save this text file in a particular location or any particular folder. I mean, couplings folder.
No, no, no. In a RNA-seq folder.
Okay.
Yeah. Okay. So now what we have to do, we need to just call the tool cuff merge. Then we are in hyphen small o, we are providing the output directory, which is output cuff merge output. This is a folder name where all the files are going to sit. Then if you want to increase the speed, you can just add a hyphen p2, that's the threads. Then in hyphen small g, we are providing our GTF file. So our GTF file name is chr16.gtf, or whatever the name you have given, you have to give the same name. Then in hyphen small s, we are providing our reference genome, because as per the reference genome and as per the GTF file, we need to be just merge the samples, and we need to merge the genes. So we require the GDF file also, and we require the reference genome also. So whatever the name you are having for your reference genome, that you have to provide. Then we are providing our input file, that's a assembly.txt. So this is what our command.
Can you once show the contents of assembly.txt?
Yes. Yeah, it's visible.
Yes, ma'am. Thank you.
Yeah. Yeah. So here the command is, and you can see the command as well as the file. You just have to run this command and tell me if you have any problem there.
Okay, ma'am.
Yeah.
It's completed.
Yeah, the command is means like the output is generated.
Yes, merge.gtf and logs file.
Yes, correct. What about others?
Ma'am, it's still processing.
Okay, let it run. So here, if you will see, like it's generating the cuff merge output, which is the merge.gtf file, and that's a logs folder. So this is our main important output file where you have all your samples merged there. So basically, this file we are going to be used as a input for the next step. And what is our next step then? Once we merge the sample, what will be our next step?
To annotate the genes.
Mm, no, but we don't have any genes right now, right? So basically, to quantify the differential expression, because once you will get the differential expression count, then then whatever the genes you are getting there, that you have to annotate. Okay?
Okay.
Yeah. So can you tell me one thing, like in which format we are getting the differential expression count? What is the unit of differential expression, or what is the format of the differential expression count?
The unit will be FPKM.
No, no, that's a normalized count
Okay,
unit.
we done previously.
Mhm. So I think in our introduction, that one concept is covered. That's a fold change. You remember
Yes.
log to fold change?
Yes, she mentioned.
Yes.
Hmm.
Yes, ma'am.
Yeah. So that's a unit. Like whenever you are getting the differential expression count, always it is in a log to fold change format. Okay. So that's what we are using the next and the last step in the pipeline. It's not a last step of analysis. This is the last step of a pipeline, which is uh, quantifying DEG, differential expression of genes using curved. But before quantifying the differential expression, we need to decide one thing, that is nothing but the analysis plan or the biological analysis plan. So, do you know, first of all, what is the analysis plan? How many analysis plans are there? Yeah, anyone?
Ma'am, like
Uh-huh.
control versus treated or
Yes.
treated versus control T.
Correct. So, differential expression or the analysis plan in the differential expression is what? Like how we want to quantify the differential expression or how we want to compare the samples that we can decide by the analysis plan. And there are two universal analysis plans we are having. One is a control versus treated and another is a treated versus control. What is the difference between that? What is a control versus treated means? We are interested to find out the expression in a control with respect to treated. And treated versus control means what? We are interested to find out the expression in treated with respect to control. Correct? So here, as for we are performing this for demo, we are going to decide the analysis plan, or we are just taking the analysis plan as a treated versus control, which means we are interested to identify the expression in treated with respect to control. Okay. So now we are going to call the tool curvediff. Curvediff. Then in hyphen O, we are providing the output directory. So that is curvediff output. Then in hyphen L, hyphen capital L, basically you need to be provide the conditions like how you want to quantify or how you want to compare, and these conditions should be comma separated, means like a control comma treated or treated comma control. This is how you need to mention. So as we have decided to take the biological analysis plan as a treated versus control, so here first we have to mention the control and then comma treated. Opposite we have to provide. If our analysis plan is treated versus control, so in a hyphen L, we need to be provide first control and then treated. Treated T should be capital. And if you have an analysis plan as a control versus treated, then first a treated and then control. So control C should be capital there. Is it clear?
Yes, ma'am.
Yeah. Then in a hyphen U, we need to be provide our input file here, that is the output of cuff merge. So you just need to be open the cuff merge output folder and open the terminal from there, because you need to be provide the path of the merge.gtf file because it is in another folder and we are running this cuffdiff from the RNA-seq folder. So open the terminal from the cuff merge output, copy the path by using pwd, and then again just give the slash, and here you have to give the output name merged.gtf. And once this is done, after that you need to be provide your BAM files, which are nothing but which are your sorted BAM files, because in that files we are having the information of the alignment. So these files also you have to provide. But if you will see that you have a 4 BAM files, right? So 4 sorted BAM files, like sorted control 1, then control 2, treated 1 and treated 2, correct? Are you getting my point? If you will see, we are having the four BAM files here: sorted, control one, control two, treated one, and treated two in your system. Correct? So,
Yes, ma'am.
hmm. So what we are going to do is we need to be provide all the four BAM files. But how? So whatever the sample first you have mentioned in a hyphen L, so that you have to provide first. So what is our first sample? That is control. Like what is our uh, first sample here in a hyphen L? That is a control. So you need to provide the sorted control one BAM file. So here you have to provide the control. But now you have a two control BAM files like control one and control two. So between the control one and control two, there is a comma. So you have to provide like a sorted control one and the sorted control two dot BAM. Then between the control and treated, there is a space. So you have to provide like sorted treated one dot BAM after giving the space. And then you have to provide the treated to dot bam by giving the comma, which means like between the control one and control two there is a comma, between control and treated there is a space, and again between treated one and treated two there is a comma. So this is our command ready for the differential expression count. I will just give a enter for now because you need to see the command properly. But it's a you just have to put it in a single line. Is it clear?
Yes, ma'am.
Hmm. Can you just try this command and keep it for the running so that it is going to generate our final output of differential expression?
Yeah, ma'am.
One here, T is small, right?
Where here?
Treated, yeah. No, yeah, here.
Yeah, actually, in my system, I have given like this. You have to give as per your system because sometimes, yeah, in your system, the name can change for the file, right?
Okay.
Hmm.
Mark, I did not get one point.
Yes.
Uh, why have you written a comma between the sorted controls and then no comma between yeah, these two? What? How are you separating them?
Actually, if you have a multiple samples of control, means like they are the replicates, right?
Right.
So that replicates we are giving by the comma. But now if you will see, this is some different samples than the control. This is not the replicate of control, correct?
Okay.
So that's what we are giving the space to
So
separate
just to
out that
separate.
sample.
Okay. Okay.
Yes. Now you have to keep this command for running so that it will generate some files inside the folder. Is it running?
Yes.
Yeah, ma'am, it's running.
Okay.
It's completed.
Okay. What about others?
It's completed, ma'am.
Hmm. Okay, so if you will see here in the folder, basically it's generated one curvediff output folder. Hmm. So the curvediff is giving us this many files. Like in your system, you will not have this Excel file right now, but other than the Excel file, you'll have all the files, right? You can see there is a file for CDS, there is a file for genes, there is a file for isoforms, TSS, promoters. So it's like the cuffdiff is not only quantified the differential expression of genes, but it is also quantified the differential expression at the CDS level, at the promoter level, at the isoform level, at the TSS level, transcriptional start site. So as of now, our interest is the differential expression of the gene. So that's what we are opening this file, gene expression.diff file. This is our main important file, and you have to open this file in Excel. So just open the Excel and browse the file. So open, browse, select the folder from where you want to open the file, make it all files, and gene expression.diff file. So just open it, then next, next, and then finish. So first and foremost thing what you have to do once you open the file, you have to save as this file in a Excel workbook format. You can give the name as a D result or whatever you want. Make sure select the format. You will select the format Excel workbook over here and save it. Okay? Tell me once it is done.
Ma'am, open gene expression file, right?
Gene expression.diff file. Yes. Yeah.
Yeah, ma'am.
Hmm.
Done, ma'am.
Okay. So here the covdiff means like this gene expression.diff file is giving us this many columns. But out of that, some columns they are generated by the tool itself. It's like these IDs, if you will search in any one of the database, you will not get any information because these are the identifiers for the tool, and we have to delete that columns because they are not for our use. So if you will see this first column, which is a test ID column, that's giving you the XLOC IDs. If you'll copy this ID and search in any database, you will not get any information about it. So just delete that. Right click and delete the column. And same about the gene ID. Okay. This column also you have to delete. Then the next column, it will give you the gene column. So basically, these are the genes which are expressed in your data, expressed in your samples. So how many genes are there? If you will see, control shift down, total 1591 genes are there. Numbers can be vary because this data is generated by like some months before, so a number can be vary. Okay. Now here the next column, this is showing you about the location, at which location this particular gene is expressed, and the location is given in a bed format, which is nothing but what? Chromosome location, start coordinate hyphen end coordinate. Then here we are getting the sample 1 and sample 2, which is nothing but a control and treated respectively, because as per in an analysis plan in a hyphen L, as we have provided first control and then treated, so that's how we are getting over here. So if you have a first control and then treated, which means your analysis plan is treated versus control. Okay. Then next you are having that is a status. So this is showing you the status of your experiment. Means like this particular column, it will give you uh, about that whether this test is successful or not. So if you are getting a no test, it means like the test was not performed, or maybe they are not getting the enough alignment for the testing. Means what usually due to the low read count or other filtering criteria, the test was not performed. Then if you are getting okay, it means the test is performed successfully. And if you are getting fail maybe somewhere, right now we are not having over here, but maybe in future if you are getting a fail, it means the test is failed due to some error or some another reasons like a statistical calculations. Okay. So this will tell you about the status. Then here you are getting the value 1 and value 2. So this value 1 and value 2 is nothing but your normalized account of control and treated respectively. Okay. So value 1 is a count of control and value 2 is a count of treated. Then in this result, our most important column is the log2 fold change column. So what is a log2 fold change? It will tell you how much amount of your gene is expressed in a treated with respect to control. Actually, how this covdiff is calculated, the log2 fold change value column, if I will show you for this one particular value. So basically here it's a log, then we are taking the ratio. So what is the ratio? We have to take the ratio of treated by control. So we are taking the ratio of normalized count of treated divided by normalized count of control and comma to the base 2 because it's a log 2 and then enter. You can see approximately same value we are getting over here, right? So this is how the covdiff is calculated the fold change value for all the genes. Okay, is it clear? Hello?
Now, if there is
Yeah, ma'am.
no test, then why it is giving these values?
No, because see, even if there is an expression, but you don't have any significance, then there is a high error there. I will tell you. Maybe you will get in a this p value column. What I want to tell you. Okay,
Okay.
this test, this status, it will tell you about the significance of the experiment, whether it is done properly or not, whether there is any error or not. It is not related with anywhere in an expression or like it is not related with expression count. Okay.
Okay.
This log2 fold change, it's an expression count, and this is what your significance of your test. Now the next value you are having, that is a test statistics. So basically this test statistics is a statistical value calculated by as they are using a t-test to calculate the significance. So we are just removing that. We don't require it. Then our next important column after the log2 fold change, that's a p-value. Okay, before the p-value, I just want to know, do you know what is the cutoff of the log2 fold change? Like because due to the log2 fold change, we can decide the genes are upregulated, downregulated, or like not expression. So what is the cutoff?
Ma'am, 1 for upregulated, negative
Hmm.
1 for downregulated, and 0 for no change.
But it's a one or like a greater than one.
Greater than 1, ma'am. Greater than
Mm-hmm.
1 and less.
Yeah. So if the log2 fold change value is greater than or equals to 1, that's a upregulated. If the log2 fold change value is less than or equals to -1, that's a downregulated. And if it's 0 or nearly equals to 0, that's a no expression. But whatever the cutoff I'm telling you, it's a standard cutoff. I'm repeating this again. The cutoff which I'm telling you is a standard cutoff. It's not a strict cutoff. You can change the cutoff as per your choice. Means like let us consider you are interested with some A gene, ABC gene, but that gene is having an expression value 0.8, and if you are giving the filter of 1, you are missing that information, you are missing the information of the gene, but you don't want the information, you don't want to miss that information because this is your key gene. So at that time what you can do, you can just reduce the cutoff, like you can make a cutoff 0.8, or maybe let us consider whenever you are giving a 1 cutoff for the upregulated and downregulated, you are getting a like a thousands of a genes, and with the help of that, like with that thousands of genes, you are not able to plot a graph or you are not able to check the interaction. So in that case, what you can do is you can just simply increase the cutoff. You can take those values which are highly expressed or highly suppressed. So that's what my point is, like the cutoff is flexible. The standard is 1, but you can change it as per your choice. Is it clear?
Yes, ma'am.
Hmm.
Yeah, ma'am.
Uh, yeah. Next is p-value. Do you know what is a p-value?
Ma'am, it's the probability that tells that how the sequences can be aligned by chance.
No, that's e-value. Hmm. P-value, if you remember, sir, we have already discussed in a microarray as well, right?
Yeah, yeah, ma'am, I got it.
Hmm. And whatever you are telling is that's a e-value in a blast. Okay. So p-value is what? It's a probability of error value, which will tell you that whether your test is successful or not, whether there is any error or not, or it is significant or not. So this p-value, it will tell you the significance that whether the test is significant or not. And to tell it's a significant, we are applying some cutoff. Like if you just filter out the significant genes, you need to be apply the cutoff for the p-value. And what is the cutoff for that?
0.005.
Sorry, what? Can you repeat, please? I didn't hear.
Is
5%,
it
ma'am.
point?
It's one. You
Zero
are saying.
0.05.
Okay, and yeah, and what is, ma'am? What you are saying? It's
Yeah, I said 0.005.
yeah, it's 0.05. Okay, it's 0.005 not. It's a 0.05, which means like we are allowing the 5% error. Okay. And
Okay.
if you will see the value which I have calculated actually about the fold change, but and you say like it's a no test, but still it is showing you the expression value. So it's not related with the expression value, but it is related with the significance because you can see no test and here you are getting a 1 p value, which means like there is a 100% error in this test, correct? So anyhow, we are not considering such kind of genes which is having 100% error, right? We need to be filter out those genes which are significant only, and we are giving the cutoff 0.05, less than or equals to 0.05. Means we are just allowing 5% error. But again, this is a standard cutoff. This is not a strict cutoff. If you want a more significant genes, if you want a highly significant genes, you can reduce the cutoff. You can give like 0.02, 0.01. The choice is completely yours. Then next we are having that is a Q value. So basically Q value is the adjusted P value you can say after checking out the false discovery rate. FDR. FDR or a false discovery rate, it's again one of the statistical calculations. So by calculating the FDR or the false discovery rate, we are getting the Q value, which is also telling you about the significance and the cutoff is the same as per the P value because this is what the adjusted P value. So we are going with the P value to calculate the significance. Okay. And the last column is a significant column, which is telling you the gene is significant or not. But that we can tell by the p value or the q value. So no need to keep this. We can delete this. So this is what our main important columns, which we require for further analysis. So
Ma'am, if
our
you change, sorry,
yeah, yeah.
if you change the log to the fold change values, then the p values will also change, right?
No. P value you can keep as it is. No need.
No, but automatically the significance will change if the fold changes, the values have changed because uh, I mean, I think that way that
No. See, that's what I'm telling. There is a no relation between the fold change and the significance. Fold change, it's an expression. The significance telling you whether the test is successful or not. So even if you will increase the cutoff of fold change or if you will reduce the cutoff of fold change, p value you can give as a 0.05 only because that's a 5% error we are allowing and that's fine means if you have a 5% error that's negligible, right?
Okay, I thought that this 5% error is calculated on the basis of these um, four change values. I mean, if you're
Uh,
saying they are independent, okay then.
no, no, actually, significance is a different and expression is a different. In expression, we are comparing how the gene is expressed in a treated with respect to control. If you remember that, I think Tanya ma'am have taken one example about the analysis plan, right? Some five genes. Yeah, if you remember, this is what the example. Uh, so this is what, like, how many reads are aligned to that particular position, and then from that we are just comparing like how the gene is expressed in a control with respect to treated and treated with respect to control. Correct? So that's what we are getting the fold change value. So
Hmm.
this is a fold change which is telling you the comparison between the control and treated. And this is what telling you about the test, the statistical test, which is calculated the error of experiment.
Okay, okay, I got it. Okay,
Hmm.
okay.
Okay. So this is about the curve diff, and our next two steps. These are the post analysis part, which is, yeah, which you are going to continue by tomorrow because obviously we need a whole session for it. So tomorrow, Tanya ma'am will continue the post analysis part. Okay.
Yeah, ma'am.
Okay. All right then. So uh, again, like once the reference based is done, on the next week, we are going to start again the de novo part, de novo analysis part. So make sure your reference based concept should be clear because the same concepts will require there, just the tools are different. Okay?
Ma'am, the test for DNA set was not given the test link.
I have given it on Monday. Monday, I think there are only four students. So,
Actually,
uh,
ma'am, we were told that there won't be lecture and there will be only test. So I thought
yes,
that the test will
yes.
be in the group. That's why I
No,
was waiting for that.
okay. No, no, no. I told you that I will take in the session. Okay, no, not an issue. Maybe let me just check 1 second. I have the link, so maybe you can just submit.
Yeah, ma'am.
Yeah. I will share the link of DNSEC test. So yeah, maybe whenever you will get time, just solve it. Okay. Whoever is not attended the session of Monday, please copy the link and just paste it somewhere.
And ma'am, was anything else taught on Monday? Was
No,
the
nothing.
okay? Okay, ma'am.
Yeah.